Hey class, this is part 2 of our video about what is DNA, and here we're going to take a closer look at the structure and discovery of that and everything we know about how DNA looks right now. Okay, so part 2 coming right now. What we know so far is that DNA is in fact a genetic material. We know that it is made up of nucleotides, which are the subunits of it. Nucleotides are made out of a phosphate group, a sugar group, and a nitrogenous base. We know that there are 4 different types of nitrogenous bases in DNA: adenine, thymine, guanine, and cytosine. And RNA has another type that's got uracil instead of the thymine. Okay, so we know that these are the building blocks of it. We know that these A's and T's come in equal ratios, and we know that the C's and G's come in equal ratios. But we don't sure about the structure yet. Here it is, really. But let's take a closer look at how we came to know that. These people deserve some recognition, so let's call out their name here. So in discovering the structure of DNA, there's some scientists. This is Rosalind Franklin. And someone she worked with called Maurice Wilkins, and they were working on trying to figure out the structure of DNA by using what is it, like X-ray photography, basically, right? Like X-ray diffraction photography. Don't hold me to that. I meant to have it on the slide. Anyway, so Rosalind Franklin, she took this photograph right here. It's a very famous photograph. It's called Photo 51. And what this photo shows, you see this like X right in the middle of it there. And this is like a photograph, sort of like looking down the tube of DNA, right? Like at a specific angle. And that shadowing effect there comes from like a crossing that can be observed through it. And if you don't know what you're looking at, like this isn't going to tell you very much, but this photo gives great evidence to what the overall structure of the DNA molecule is going to look like. So this is a very famous photograph, and it's worth noting. This is Photo 51, taken by Rosalind Franklin. All right. So then let's put the whole picture together, right? Watson and Crick, these are the 2 guys that credited with sort of like discovering and modeling a DNA molecule, figuring out the full structure of it, taking the evidence from everybody else. The photograph from Rosalind Franklin suggested the double helix form. The information about nucleotide, just knowing that it's nucleic acid from the previous scientist, knowing that the DNA is made out of subunits called nucleotides, and that these nucleotides come in equal ratios. Chargaff's rule here comes with a very useful bit of information about these equal ratios. If you notice, these A's and T's, they have sort of like complementary shapes, right? A's have these 2 rings, T's have 1 ring, G's have 2 rings, C's have 1 ring. So there's a nice like continuity in this. Ultimately we're going to call this base pairing. What's going on here? Okay, but these guys came up with the double helix structure. Where we see specifically now here that these nitrogenous bases are bonded together, that it's always in the same grouping, right? Chargaff had the evidence that they came in equal numbers, and then these guys put it together, and probably others too, but that these are meant to fit together. The picture from Rosalind Franklin suggests this double helix, and so we have this double helix. Helix means it's twisting, right? There's twisting action going on here, and so there's 2 of them. And so if you were to look down this molecule right here, right? If you were to look down this, you would see that X shape that she took that picture of. So they took all this together. They figured out, like knowing the structure of this, they figured out that these phosphates and these sugars have a favorable interaction where they're going to line up, and we call this backbone of the molecule, right? And so this happens on both sides. We have these 2 backbones, and then reaching across and binding in the middle are these nucleic acids. You can see the 2 rings and the 1 ring, the 2 rings and the 1 ring for the purines and the pyrimidines. Okay, these are held together with hydrogen bonds. A lot of hydrogen bonds putting it all together. For you chemists out there, you know that hydrogen bonds are overall really weak bonds, so they can be broken easily. And that'll be helpful as we try to figure out the function of this molecule. We talk about making copies of the molecule or trying to transcribe it in order to create a protein and try to read that code on the inside. But we also know that hydrogen bonds, when they're in great number, they're very strong. And DNA is a really big, long molecule, so there's a lot of these. This is a teeny tiny segment right here. There'd be billions of these pairings going on throughout the molecule. So it's a really, really big molecule here. Okay, so lots of hydrogen bonds creates great strength and stability. All right, so this is the whole picture that we've got, right? They also gave us information about the direction of this. There's a specific direction in which the molecule can be read, depending on which side you're looking on. Okay, you can see the arrows here and here. So we call this the 5' end and the 3' end. And where that naming comes from, it has to do with the sugar molecules that are on here, right? This is a pentose, right? So this is a five-sided ring, like a pentagon, and so we number each one of these, okay? So, you maybe go like 1, 2, 3, 4, 5, okay? So this 5 right here, and then we come down here, and it's 1, 2, 3, okay? So we got like 5 side and a 3 side. So it's just, they were numbered, there's a specific way that they're numbered, and if you're a biochemist, you'd know how that's supposed to go, so in the future, maybe you'll understand that better. But for now, we just need to understand that it's being read in a specific way. Whenever you see a DNA molecule, it'll be labeled as a 5' end and a 3', so we'll know which way that it's supposed to be read. And then these run in opposite directions, right? So if one of them is going 5 to 3, then this one's going 3 to 5, but always being read in a specific direction. Some useful stuff to know about the shape of this molecule. We know that the code, the actual information that's being stored on the inside. That's the sequence of these bases that we have running throughout here. Okay? We know that because of the backbone that we have here, this gives it a lot of stability. And so this is going to be a heavily used molecule over and over and over again. And so we need stability. We need it to be strong. So we can learn a lot about the function of this and based off of looking at the structure of it. So Watson and Crick are the guys who famously gave us that information. Okay, so a little bit more then. Let's take a look at just DNA and some of the forms we can find DNA in. We'll look closely at these, but here's what we know of DNA, basically the backbone, the pairing, the double helix that goes along with it. Okay, but there's an organization to it when it's inside the cell, and so there's some different forms of DNA. We needed to look in these different ways at different times based off of what it's doing. Structure equals function, right? You got to change the structure, the shape, the way it looks if you want to do something different with it. Consider maybe something as simple as just like a piece of paper, depending on what this piece of paper looks like, you might do something different. You could write on it, maybe you could hide some information in it, you could fold it into an airplane, you could crumple it up and shoot paper baskets with it. Depending on the shape of it will affect what you can do with it. So DNA, we have a couple different ways we're going to look at it, right? We have DNA right here with the sugar phosphate backbone and the purine pyrimidine base pairing. We've got the double helix shape. We've got this DNA wrapped around proteins, start to condense it there. Then we have a continuing twisting and coiling, making it a tighter and tighter package into what we call supercoils. And then ultimately a really tight, dense, compact DNA in the form of a chromosome. So DNA is in all these different forms at different times, depending on what it is you want to do with it. Okay? So here's our regular old DNA. I think I've talked enough about this for now. We'll move forward. Let's talk about DNA and going from DNA to chromosomes. We have this thing called the nucleosome. And this is where that DNA starts to wrap around proteins that are called histones. Okay? And so all that's really going on here is that we're trying to store the DNA for later use. right? If nothing's happening to it right now, we're not using that specific piece of DNA, so we're going to wrap it up, fold it up, and store it away for later. So this is a really convenient way to do that. Because of the shape of it, the spiraling, it offers the ability to move and wrap around these proteins in a certain way. Okay, so this is what we call a nucleosome, DNA wrapped around these proteins. We can continue to condense it, and we look at DNA in the form of chromatin. Okay, so DNA spends most of its life in the form of chromatin. And so here, just a different look at that progression. Regular old DNA into the histones, and then as we tighten that up, we end up with what's called chromatin fiber. Okay, so typically when you look at a picture of a nucleus in a cell, whether this artistic one or an actual picture, this DNA is being stored as chromatin, not chromosomes. DNA spends very little time in the form of chromosomes. It's a specific time for that to happen. Okay, so think of this as like a bowl of spaghetti, okay? It's just a big mess. It's all twisted and all over each other. It's a bit organized because of the histones and the tightening that goes on here, but overall the DNA, it's not organized. It's just all twisted and folded on top of each other, like a bowl of noodles, you know, or I don't know, like gross you out, like a snake pit, something like that, you know. But it's just everywhere. This is how it spends most of its time. Okay. So remember this picture right here, because we're going to look at a picture of DNA as chromosomes here. So here, take a look at this picture. Right. This is an artistic drawing of a chromosome, and this is how we see it so often in textbooks or just like pictures online. Like when you talk DNA, we see like this part right here. We see this picture, or we see this, and this picture is cool, it's artistic, but it's very misleading. Okay, so you know, like here's chromosomes, and this is what we call a karyotype. This is a map of our of a human chromosomes. Okay, and there's 23 pairs, and we'll get into that part of it later. But you know, like these are common pictures we see, but it's misleading because DNA doesn't spend its time like that most of the time. Okay, most of the time it looks like chromatin, which is not this, right? It's not this thing right here. That's not chromatin, and it doesn't spend most of its time as this. There's all sorts of stuff that's misleading about this picture here. So here's a picture of some cells at various stages in their cycle, and here we can see the chromosomes here. Okay. Most of the time, a DNA, it's going to be looking like these ones here. Okay, this is chromatin. Here we see a cell at the beginning of what we would call prophase in mitosis, where that chromatin, it's starting to condense and it's starting to form chromosomes. Here we see it probably in like metaphase, where DNA and the chromosomes are being separated, but you can clearly see the individual parts. Okay, most of the time, regular function is going to look like this. Okay? So that's pretty much it for the structure of DNA, and we're going to look at, again, like how all those different structures are going to affect function overall. Okay, this picture is a great lead into when we get into the function. This is a molecule called polymerase here, and there's a lot of function going on here. So I hope you guys like this video. See you in class.